economics. There we go. And I also direct the Center for the Study of Public Choice and Private Enterprise. And I'm also currently the president of Phi Kappa Phi. So thank you all for being here. So Phi Kappa Phi, just to give you a little bit of information, it's not a fraternity or a sorority. It is an honor society. Membership is not granted to all who maybe desire to be in it, but it's something that has to be earned. The Phi Kappa Phi members at North Dakota State University are among the highest and brightest achieving students in the nation, and they typically represent about 3% of the university's student population. The Phi Kappa Phi faculty lecture was established in 2015 to recognize faculty whose outstanding high-impact research, scholarship, or creative activity consistently supports NDSU's top-tier research status and have a proven ability to effectively present their research to a broad community in the land-grant tradition. Tonight, we'll congratulate the winner of this year's Phi Kappa Phi Lecture Award, Dr. Erin Gillam. But before we do that, I want to recognize the two finalists that were also selected by the committee. Dr. William Ganja, Professor and Chair of Agribusiness and Applied Economics, and Julie Garden-Robinson, Professor of Health, Nutrition, Exercise Sciences, and Extension Specialist. I also wanted to thank Jean and Bill and Green, Vice Provost for Faculty Affairs and Equity, who facilitated the selection process and arrangements for tonight's presentation. After Dr. Gillam's presentation, there will be some time for Q&A, and then we'll have a reception following as well. And please welcome Dr. Kimberly Wallen, Dean of Science and Mathematics, to come make the introductions.
All right, good afternoon, everyone. Thank you for joining us on this great lecture this afternoon. So as stated, my name is Kimberly Wallen, and it is my absolute pleasure to introduce this year's PKP Fellow Faculty Lectureship. I'll slow down here in a minute. Dr. Erin Gillam. Erin is an outstanding member of our biological sciences faculty, our college, NDSU, the community. I really can't say enough about her. After earning her PhD at the very prestigious Department of Ecology and Evolutionary Biology at University of Tennessee, she went on to do a postdoc at University of Virginia, and then she joined NDSU. We were lucky to have her join NDSU in 2009. Since joining NDSU as a behavioral ecologist, her research on bats, and by the way, bats are awesome. I'm just going to really show that bias. Her research on bats and the conservation of bats is highly relevant and highly respected, both in the research community, but also in the state of North Dakota and the many stakeholders that we have in the region and the world. Much of her research is conducted as experimental research in a field setting. And for any of those of us that have done that, know that that is very challenging. There are a lot of things that you cannot control, but I'm sure she'll talk about some of those. In addition to that, she has mentored over 15 graduate students and worked with 30 plus undergraduate students in her field research. So they're getting to be exposed to and learn that doing experimental field research is challenging, but it's also a really unique opportunity to see bats and animals in their natural setting. So it really shows them a very robust amount of data and things to think about. She's published over 45 peer-reviewed journals, which have included other colleagues, as well as graduate students and undergraduates as co-authors. She's been invited to numerous presentations about her research. And what I think is really impressive is that she's given over 300, maybe it's 400 presentations to the general public. And that really speaks to her ability to translate complex research that she's doing in the field to the general audience. And with that very brief introduction, like I said, I could say a lot more. Please join me in welcoming Erin to the stage.
Hey, could you help me clean up all this garbage on the screen? Maybe first?
This one doesn't show up. Oh, that is.
Okay, I'll make that one go away too. Okay, thanks everybody as we navigate some Zoom figuring out. So can everyone hear me okay? So thank you so much for coming today. Before we get started, I just want to thank my Department of Biological Sciences, our Faculty Affairs Committee that wrote the nomination letter for me, as well as the University Awards Committee that makes the selection, and Jana Beeling-Green and Ann Fredrickson, who were really critical in putting this event together. This has been one of the hardest and one of the funnest talks I have put together in my career to date, because I could talk about like anything that I've done, right? And like there were so many potential paths of nerdiness that I could take you down that it was really hard to decide which one. So hopefully what I've stitched together kind of gives you a picture of some of the things my students and I have been doing over the years since I came to NDSU in 2009. Okay. I gotta click. Hold on. Okay. Nope, you guys can't. All right. It's a little slow. So before I get started, just want to give you a brief outline of some of the topics I'll be talking about today. First, I'll start talking about echolocation, which is a sensory system that's really critical to bats for how they see the world. I will note that I stole the title of my talk from the famous Don Griffin, who wrote the book Listening in the Dark. This is the person who really first effectively described echolocation in bats. So I'll talk to you about a little work I've done on that over the years on echolocation. Then I'll get into talking about social communication in bats, specifically about some work I've done looking at how bats use social calls to maintain contact with other members of their group. And then finally, I'll talk a little bit about work we've done here in North Dakota, where we listen for bats, listen for their echolocation calls, and use that as a tool for conservation. Bats face a lot of threats around the world and right here in North Dakota. And so this is kind of a powerful tool in our conservation toolbox. Okay, so let's just jump into echolocation. We're a little bit slowly. There's a little bit of delay. So echolocation, as a refresher, or maybe you don't know, is an active sensing system in which a bat produces a generally loud call that is broadcast in the environment. That call bounces off objects in the environment and produces weak echoes that return towards the bat. In this case, in this little diagram, this is like a tasty little moth that the bat might want to eat. And there's a weak returning echo from that moth. And not only can the bat detect this returning echo, it can actually extract a huge amount of information about that target from this little echo, right? I'm going to show you a video. It might be a little slow. We'll see here. And this is going to be of a bat that is pursuing a moth that is kind of tethered to a line. And you should be able to hear the audio. The whole thing is slowed down, so the bat's moving slowly, and the audio is slowed down so you can kind of hear the echolocation calls. Oh. Zoom is struggling. Okay, so this will kind of keep playing in the background, but what I want you to take away from this is that even just by listening with your ear, you can tell that those echolocation calls are changing a lot as that animal is moving from further away to when it's getting really close and then making a capture attempt. I think at some point it does miss the moth, actually. So echolocation is a really dynamic system that the animal can change very rapidly to kind of optimize it for the type of information it needs about its surrounding environment in that given moment. If we were to look at these same calls like on a computer, this is what we would see. And I'll just kind of orient you to these images because you're going to be seeing a lot of these graphs. This is a spectrogram, which is time on the X and frequency or pitch on the Y. And this gives us a picture of what these calls look like on this time frequency scale. And what we see is that, you guys can't see that. Is that these calls change a lot. Again, we go from like what we call search phase, where the animal is searching for a prey item, to once they detect something, you can see the calls get a lot more broadband, which is much better for getting really specific information about the location of the target. And as it gets closer and closer to the insect, the calls eventually kind of end in this like kind of sound that's called a feeding buzz. Okay. So again, this just highlights that this is like a 2-second sequence. These calls are being modified by the bat from like call to call, basically very rapidly to accommodate the information needs of the animal. Now, one thing that I've been interested in for a long time is understanding how echolocation operates in the real world, right? So bats are not echolocating in a vacuum. They're in an environment in which there are potentially sources of acoustic interference that could mess up their ability to detect those weak returning echoes. And while there's lots of things that could potentially do that, one I've particularly been interested in is what happens when bats are foraging in the same space as other bats, specifically bats of the same species, because bats of the same species produce pretty similar calls. And what happens is that there's a problem called acoustic masking. So what happens is that, let's say our focal bat emits this, the black call is the original echolocation call, and it has to pick up this weak returning echo in gray. But if another bat comes by, we are having problems here. Their loud calls may overlap that weak echo, right? And once this happens, this kind of interferes with the bat's ability to effectively get the information it needs from the echo. And again, this is called acoustic masking and should be a problem for lots of bats that end up foraging with conspecifics in the same space. Because I don't like to do things in extremes. I mean, not in extremes. I'm going to talk to you about some work I've done with a species that has this problem kind of in spades, right? So this is the Brazilian free-tail bat, Pteridata brasiliensis, also sometimes called the Mexican free-tail bat. It's the same species. And they are found in they have range from like the middle United States down to parts of Mexico, and there's some subspecies that do in fact go all the way down to Brazil. I have studied these bats in Texas. And why are they so interesting for thinking about interference amongst conspecifics? Let's see if I can get this started. Okay. So this is an emergence from a cave that is a couple miles from where I've done my field work in Texas. And there's estimated to be about 2 million bats in this cave. And so we're not even going to talk about the sensory nightmare that is trying to like navigate in that like thing of like thousands of animals. I want to like zoom out to after these bats have dispersed across the landscape and start foraging for insects. They're going to run into other bats of the same species, and they're going to have this potential issue with acoustic masking. So what do bats do when they face this problem of potential overlap with the calls of conspecifics? Well, we can talk about this concept called jamming avoidance responses, which is a suite, I'll eventually get to this, is a suite of behavioral strategies that animals can potentially use to avoid overlap with the echolocation calls of other bats. So I'm going to talk to you specifically about frequency or spectral jamming avoidance. And I am really like, again, I could kind of decide how I was going to take this talk. And so like, we're going all the way back to my PhD. And like, I kind of chose to do this because I feel like for this talk, I can talk about a bit of the arc of my career. And I haven't done a lot of this echolocation work in a long time, but I'm starting to do it again and kind of getting back to what some of the same sort of questions I asked in my PhD. So in the mid 2000s, people had not really investigated this idea of jamming avoidance in bats too much. There had been a variety of like correlation studies where people had recorded bats when they were flying alone and saw that the frequencies that they were using were pretty similar when you compared them. But when they had bats flying, like two bats flying in the same airspace, they found that there was greater frequency separation. Right. So it was a correlation, but it seemed like how you kind of see here when the animals were together, they were making a behavioral shift in the frequencies they were using, potentially to avoid frequency overlap with each other. But again, this had not been done experimentally. So for my PhD, one chapter of my PhD was the first experimental test of jamming avoidance, specifically spectral jamming avoidance in bats. And I'm not going to get too much into the details here, but I'll tell you just a little bit about the experiment I did, which mostly involved sitting in a corn or cotton field by myself at night and messing with bats with playbacks. As Dean Wallen mentioned, I really like field experiments. And so I used a playback system in which I could play back echolocation calls of bats to them in the field and saw how wild free-flying animals responded to the sounds that I played. So just to give you a little bit of background on what this experiment was, I had a sequence of calls from this bat species that I had like experimentally sort of like increased in frequency and decreased in frequency. So I had this like whole bunch of playbacks that were the same, except they were shifted up and down in frequency. And I would do this thing where I would have a signal, the pre-switch playback, playing for some length of time, just like waiting for a bat to come along, right? And the bat would come along. And then as it got closer and closer to my setup, I would switch it. Right. And so I would switch it to a different signal of a playback of a different signal. Right. Now, some of these, I had all sorts of frequency switches and combinations, but the ones that were really interesting were the ones where pre-switch, the frequency separation between the playback and the bat was large. Right. So here you can see the red is the playback and the blue is the bat, and they're not really overlapping very much frequency wise. But then I bring this playback signal down, and now it is right up in the same frequency space that bat is using, right? So I've really introduced a potentially interfering stimulus into this one animal's sequence, right? Like, luckily, this is a great site because the bats would kind of come along one at a time. It's like perfect for sampling. So what do we find? Again, trying to figure out like, do these bats adapt their calls when they are faced with potential acoustic interference and acoustic masking from the calls of conspecifics? Well, what we found was that the bats really rapidly changed their call structure in a way that reduced overlap. Okay. Interestingly, they tended to mostly shift their call frequencies upwards. And this would happen very fast. We often saw reactions in as little as 200 milliseconds. The animals would change the structure of their echolocation calls once the interfering post-switch playback was introduced. These are just two examples here. So here, the pre-switch, when I say frequency here, I'm talking about like the minimum or lowest frequency of the call, just because that's kind of easiest to describe. But the frequency here is really quite distant from the one that the bat is using. But then post-switch, it's really like right up in that same space. And the animal makes a subtle, but switch there to potentially avoid as much overlap, right? Here's another example. Sometimes we didn't have a pre-switch, we just had silence. And in this case, we introduced the post-switch again, right in that same spectral space. And we have the signal, again, the animal is increasing the frequencies, which aligns with what we would expect if this kind of jamming avoidance response is happening. Now, it's been a long time since 2007. So there's been a lot of experimental work done on this idea of jamming avoidance in a variety of other bat species since then. And so we know a lot more about this now. Like I said, I had described the frequency shifting in the Brazilian free-tail bats. We also know in the same species that sometimes they just reduce how much they're calling, right? So they give fewer calls. And if like all the bats do that, then like there's less chance of overlap, right? In terms of like temporal overlap, if they're just calling less. We also have this bat, the big brown bat, that has been shown that in like a lab environment, when two individuals are flying together, specifically when they're in like a leader follower position, that the follower will stop calling completely, which is like super fascinating, I think, because it's like, are they trying to avoid overlap, or are they trying to like use the pulse echo pairs of the bat in front to do something? Like, we don't know, right? But it's they seem to stop in the presence of other bats, at least for a couple of seconds. In this species, Kuhl's pipistrelle, it's been shown that the bats, when they are flying in the presence of conspecifics, their calls are longer and louder, which is kind of a typical mammalian response. You know, if it's a noise in the background, you speak louder to kind of get your person to hear you. And then there's been work on this greater mouse-tailed bat that's shown that the bats don't actually exhibit a jamming avoidance response at all. That somehow it seems they're able to parse out their echo, even if there's overlap. And they make some changes associated with the presence of another bat, but they make the same changes if there's like a tree or something, right? It's like just another object in the environment. They don't seem to be doing this kind of at least spectral jamming avoidance that we've seen in at least a couple other species. So that's where we're at. And I'm gonna return to this at the end of the echolocation section. But before we do that, I want to talk about another area of echolocation research that is a little bit older in my career for me, but I'm coming back to as well. And I will talk about what I'll be doing in the future. But this is also about flexibility in bat echolocation, that this is a highly dynamic system. But now I want to talk about altitudinal differences in echolocation. So the same species, Brazilian free-tail bats. Let's see if this works here. So what I'm showing you is a Doppler radar of South Central Texas. Those orange circles are the major cave colonies of bats in this area that can have anything from a couple hundred, maybe couple tens of thousands to a couple hundred thousands to a couple millions, right? And so you can see that as the bats emerge nightly, they are picked up on the Doppler radar, right? And this is sort of a clue. There's other lines of evidence too, that the bats are also not just dispersing across ground level, that they are going up high, okay? Based on some research by my colleagues, so I don't have time to talk about, we know what they're doing up there, okay? So migratory moths often take advantage of prevailing cold fronts, particularly in late summer and fall, and they use those for migration. It's just like kind of free ride, right? These tend to kind of be concentrated around 400 to 600 meters above ground level. And the bats are going up there and feasting, right? So the bat on the left, you can see is sort of a normal mass Brazilian free-tailed bat. And this is one that was caught the day after one of these cold fronts had come through, right? Where they knew they were really up there eating, right? That bat is lit. I mean, it's double the weight, right? Look at his butt. So they are really, this is an amazing source of food for them, especially in the fall when other sources are kind of going down as it's getting a little bit colder. So getting back to echolocation, one thing we were interested in was understanding, do they change their echolocation calls with altitude, right? They're going up high, like over 1,000 meters, right? And things change there. So like one thing is that the atmospheric conditions change, and things like temperature, relative humidity, and air pressure, we know change patterns of sound attenuation or degradation in the environment. And we also, the environment is different up there, right? We don't know, like the distribution of the insects might be different. They don't have to deal with potential obstacles like they do at ground level. So there could be a variety of factors that would potentially be driving change in echolocation across altitude. So this is work that happened before, I wasn't involved in this, but what my PhD advisor, Gary McCracken, and his colleagues did was that they put bat detectors, which are machines that can detect and record the high frequency echolocation calls of bats. They strapped those to a kite or a balloon and put those up high. And by doing this, they were able to make recordings of bats up to about 1,200 meters. And that was not like a bat limitation. That was like an equipment limitation, right? And here are just some exemplars of what they saw, right? So at different altitudes when we analyze these echolocation calls. What you can see is that from when we go like close to ground level to much higher up, these calls are getting, they're getting longer and they're getting more narrow band, okay? And specifically, those types of changes are associated with calls that will travel further in the environment. Now, I will say that these changes are way more than we would expect just based on those like changing atmospheric conditions alone, right? So there's got to be something else going on here. And honestly, we don't really know what it is. This is still an open question. I mentioned that the density of the moths tends to peak around 400 to 600 meters above ground level. Maybe up at like 800-900 meters, the moths are spread out a lot more. So they need calls that can detect more distant targets. We don't know, still an open question. So coming back to this has all been on my mind, which is why I wanted to incorporate this into my talk, was that I am starting to revisit questions about jamming avoidance and the high altitude work as well. In my work. This summer, I'm planning to go to Texas to do a study. I mentioned that leader-follower behavior that was seen, that the bat in the back turns off their echolocation. I want to look at that in these free-tail bats and see if there's, when I'm doing a playback, right, do they preferentially approach the playback from the front or the back? Because that will modify the types of acoustic interference they're receiving and the kind of information that they can gather. What I'd really love is like a bat on like a fake bat on a string that I could make go back and forth and move, but like I haven't quite figured that out yet. But the technology has come a long way since I was a grad student. I now have a multi-microphone array, which is what I've kind of made a diagram of here. And this allows for not only recording of echolocation calls, but you can reconstruct the 3D position of the animal based on that call, and then get a flight path and match that up perfectly with the echolocation. So it allows you to get a picture of the animal's movement, which is its behavior, right? Its behavior in space, and couple that with the types of echolocation calls it's producing. In fall 2021, I also started a project with a couple of colleagues that was, again, revisiting the high altitude stuff. And again, we were just able to record the bats previously, but we don't know what's happening behaviorally up there. So this is one of these on a kite, on a balloon string, right? Basically. And we were testing this system, ran into some problems the first time in fall 21, but we're going to continue to work on this. And the idea is that if we can get this multi-microphone array up, then maybe we could figure out what kind of movements and behaviors the bats are engaging in when they are calling and get a better picture of that. Okay, so just to sum up that, I swear the other sections are not as long. You won't be here all day. The main thing here is that with these Brazilian free-tail bats, but really with all bats, flexibility in echolocation is really critical for them to adapt their echolocation calls to meet their information needs under different behavioral and ecological conditions. And I think the Brazilian free-tail bats are like extra flexible, but this should be something that's important in all bats and has been shown, you know, most bats are really highly flexible in their echolocation. Okay, so let's switch to sociality in bats. So bats, there's over 1,400 species of bats, okay? And they vary a lot in their social structure. Bats are highly social animals, right? And so if you look across those 1,400 species, you see a lot of variation in key describers of bat sociality. For example, group size. You have everything from this red bat over here that mostly leads a solitary life and really only comes together with other animals to mate, all the way up to those Brazilian free-tail bats I talked about, where there's millions in a cave. We also have quite a bit of variation in the way animals are socially associating specifically in terms of who's mating with whom, right? We call these mating systems. We have everything from this bat down here, this gorgeous carnivore is monogamous, right? To a lot of bats that tend to like meet up at central locations in the fall, and they do what's called swarming, usually outside a cave or a mine, and they are mating in flight, right? And that would be promiscuity because they're often like mating with multiple animals in a night or across nights, right? And then you have some other funky ones where the males are defending female groups. This guy like honks, and then the guy who honks the best and is most close to the middle of the group gets the most mates and things like that. So tons of variation in these mating systems. Now, I've always been super interested in bat social groups, specifically in understanding like what kinds of information are they exchanging, okay? As well as how do they stay together? Okay, bats kind of vary in how stable their social groups are, but often they do stay with the same individuals for extended periods of time. And how do they go about doing that, right? Because they fly at night, right? And so you have to be able to relocate your group mates. So like what mechanisms are permitting that group stability? And I would argue that one of the most important of these mechanisms is communication. Okay. And again, because it's nighttime, they tend to not use visual stuff as much. We're talking mostly about acoustic communication. So I want to dig a little bit more into this idea of group stability, right? So for a lot of animals, including bats, they live in a permanent or semi-permanent structure, right? The group lives in this refuge that's their home, right? And so if this is the case, it's not such a big deal when individuals go out on their, come on, bees. When individuals go out on their own to forage and disperse across the landscape, as long as they can, as long as they have the spatial memory to know where that central roost or refugia is, they don't have a problem relocating the group. But there are lots of situations in which there is no central permanent place that the animals live. Some groups of animals move around, right? And don't have like a set place that they are always living. And other animals use really ephemeral places to live, have really ephemeral places to live that they have to change really regularly. And so my collaborators and I were interested in understanding what role communication could potentially play in situations like this. Now, we suspected that there was a role because in other species, particularly in mammals and birds, there have been things called contact calls that have been described. Contact calls are social calls that permit and maintain group cohesion in a social group. These are some examples from a couple primates and a bird. And again, these are given in situations where that permit the group to stay together. You can see there's even some kind of specialization of them. For example, in this capuchin, you've got one call type that's given when one animal is like kind of separated from the group, and a different one that's given when there's like the group is going to begin to move, essentially. So clearly in some animals, communication is an important mechanism for maintaining, group cohesion. So again, extremes. So my collaborator, Gloriana Chaveri, and I were interested in understanding this idea of the importance of contact calls in bats. And we looked at a species that has particularly ephemeral, short-lasting roosts, okay? This is Spix's disc-wing bat, or Thyroptera tricolor. You will notice that it gets its name from the fact that it has suction cups on its wrists and ankles that it uses to adhere to the smooth surfaces of not plastic, which is here usually, but leaves, okay? So these animals live in the furrowed leaves of heliconia and calathea plants, also like banana leaves sometimes they'll live in. And just to give you an idea, if you've ever seen a plant like this, right? This is like an immature leaf that is maturing. And it starts out as like a really tightly rolled tube that like nothing can get into. And then over the course of like 2 to 3 days, it slowly opens until eventually the whole thing is just open in a flat leaf, right? And so during a period of time, the openness of that leaf is good for these bats to live in. And so that's what they live in. They are found in Central America. I have done work with them in Costa Rica with collaborators. They tend to form smaller groups, like usually like 3 to 6 is kind of the more average numbers that you get, but they have really stable group associations. I'll show you, this is my collaborator, Gloriana. She has done a lot of work on this species as well, some with me, some not with me. But she had done some work that showed the stability the stability of these group associations is really amazing for bats. The same set of individuals in a group will stay with each other for up to 2 years, okay, which is a long time in bats. So these are very stable social groups these animals are forming. But at the same time, this roost, these leaves they're using are only good for 24 hours, for one night, right? So every night they have to find a new roost. Now they are loyal to a smallish home range, right? But within that home range, there's multiple patches of leaves, multiple potential leaves that they could be in, right? So they still face this problem. If they get separated from their group, they can't just go back to last night's location to find the group, okay? Because that leaf is no longer suitable as a roost. So coming back to field experiments again, Gloriana and I did an experiment in which we wanted to see if contact calls were potentially playing a role in reunion of individuals from the same group when they were separated, right? So what we would do is we would catch a social group of animals that were all in the same leaf together. And then we would set up our experiment and we would have a suitable leaf like this one that we would set up, and we would put one of the bats from the social group in here. And like, this is like one of those things that's like really lucky with methodologically, like the bat would stay in there, which was like, wow, I don't think we could have done the experiment if the bat wasn't just like chill in the leaf, right? So we put the bat in there, and then a couple of meters away, we have another bat in the hand that we release. And these things are all just our microphones that allow us to capture any sounds that are produced both by the flying bat and the bat in the leaf. And perhaps not surprisingly, we found that there were contact calls here. What was kind of surprising was that it was not, oh, I'm sorry. Let me show you this first. I forgot about this is like the worst video, but I'm going to show it to you anyway. So this is the suitable leaf, right, that we've set up. And if the video plays properly, you'll see the bat kind of fly by, and like I've released it just a moment before that. I'm going to show. There it goes. Did I freeze it? There's such a delay. Here it goes. Okay. Okay, so that was a little delayed, right? But that was like less than 5 seconds or something like that. And you could say like, I mean, it's the only leaf right there. Of course it goes in there, right? But that's still pretty fast for that kind of assessment, right? And so when we looked at this, we did in fact see that there were social calls being produced. And what was surprising was that there was not just one social call, but 2, and they were produced in a very specific order. So first we found that the bat that was flying, the one that we released, would produce an inquiry call, which is this one up here, right? And then very quickly, the animal in the roost would produce a response call, okay? So these response calls were, first of all, the response was very rapid, right? Often from the time the inquiry call ended, it was like less than a second, and the other bat the bat in the leaf would start calling, right? And the bat in the leaf would also give multiple of this. It would emit like a series of calls that the animal would, like, whereas this was just kind of like one at a time. This was like a bout. So I do want you to notice for later that this calls, in terms of acoustic structure, is pretty simple, right? Whereas this one, it's kind of hard to tell, but these are like little U's. And like, so there's syllables in there that are building out the call. So this is quite a bit more complex than this, right? But overall, they were clearly producing these calls. And so this contact calling system, this sort of call and answer system, very much appeared to be playing a role in how they find each other when they are separated. And I think there's a lot of reasons that they could get separated, right? Not only if they're flying at night, but like funky large mammals or researchers come by and like, it's like a leaf. So like it can get bumped and the group's flushes, right? So I think there's a variety of conditions under which this could happen, that one animal is in a leaf and the others are looking for a leaf. Basically, they're looking for their group. The next thing we did was look at whether these calls had individual signatures for both the inquiry and the response call. And this involved recording a lot of individuals and individuals within different groups, essentially, to see not only is there an individual ID that's kind of encoded into this, but also a group ID. We found for both of these call types that there were strong individual signatures. So essentially each bat was producing its own unique variant of this call, right? I will say that they were really strong for the response call. They were strong here, but like these are like really different between individuals, which makes sense because again, this call is more complex. You can think about it that there's more acoustic space for individuals to carve out their own little signature, whereas this one's not as complicated, right? But we didn't really find group signatures were a lot weaker. So it seemed like, and you remember these groups, there's like relatedness within these groups too. So like the groups, we couldn't really suss out the difference there if it was, if there was a genetic piece there too. So weak group, but definitely individual signatures. Now, just because there is like statistical differences between calls of individuals doesn't mean that the bats can detect that or that they actually use it in behavioral decision making, right? So we kind of drew from this that the differences, individual call signatures were there, but further work was needed to really see if the bats were using that information. So we did experiments in which we tested the bat's ability to discriminate group from non-group calls, right? So in the first experiment, we took a bat and put it inside a leaf and then broadcast calls nearby, inquiry calls that were either from a group member or a non-group member. And this wasn't like a neighboring group. This was like a group from like another site far away. So they had no familiarity with the individual that produced these non-group calls. Okay. And if they are discriminating and preferring to attract group mates, we would expect stronger responses to the group inquiry call, the calls from a member of the group compared to the non-group. Come on. That is not what we saw. Okay, so these bats that were in the leaf, they would, if they were going to produce a response call, they would call to anybody. Right? So it did not matter if they were, if it was a group member or not, right? We don't know why this is, because it could be that, remember, this signal isn't as complex as the other one. So maybe it's harder for them to tell individuals apart, or maybe they simply don't care. Like maybe there's benefits to being in a group, even if it's not a familiar individual. And so they'll call to anybody. We don't know. Those are just two possibilities. We can't tell the difference between those. We also did this experiment the opposite, right? Where we had a flying bat, 2 leaves, and from one leaf was emitting the response calls of a known individual from their group, and the other was an unknown individual, right? Here we did see a difference. Okay, so they did sometimes go into the leaf of the non-group caller, but more so they went into the leaf of the known calls that were coming from, right? So this is indicating that they are able to use this information when making behavioral decisions about which leaf to go in, right? So at least for this one call type, again, this is the more complex call type, so maybe it makes sense that they can do this for this call type, but not the other. Again, I'm just hypothesizing, I don't know. But in this case, they can make that discrimination. I just wanted to talk very briefly about a couple of other studies that my students and collaborators and I have done over the years. My former PhD student, Karina Montero, she was really interested in the social calls that the bats were producing at night when they're flying around. And she described this call called the LUM call, which it just has to do, it's like upward modulated or something, it's like based on the structure of the call. But this is a social call that she really commonly recorded when the bats were flying around, kind of, in the same airspace to some extent, right? Like not right next to each other. So we thought that this may be an additional contact call that they use when they are in flight and kind of on the wing, rather than when like they're searching for a group member in a roost already. So it seems from what we saw with this that there's potentially multiple types of contact calls they're using. Gloriana and I did an experiment in which we tested the effects of the leaf structure on the sound, okay? Because this is essentially like a horn, right? That the sound is either coming out of or going into. And we wanted to see how this changed the sound. What we found is that in this condition, the incoming sounds were really quite amplified, right? They got a lot louder as they went in here. It's a little bit louder here because of directionality, but this was much stronger. But what we did find was that both types of calls were distorted to some extent by the presence of this leaf, right, being in the way of the transmission between sender and receiver. And again, this could potentially be part of the reason that we don't see responses with that more simple inquiry call if this is kind of muddying the water, again, with these individual signatures with this amount of distortion. And then I'll show you this video too. This is a video that Karina collected. Again, she was interested in kind of how the bats were also like assessing unoccupied leaves. And so this is a leaf that has not been used before. And that's a group of bats. And this is like the middle of the night, right? And they do come out again at some point, right? So it's not like they're done for the night. They're kind of like assessing the leaves in the landscape. Yeah, like there they go, right? So what this suggests is not only that there's some interesting inspection behavior that is involved in them picking a leaf roost, but also that for at least some of the night, they're spending time together. And Karina actually used radio telemetry systems to see how much time these animals were spending kind of in the same area roughly. And she found that this was much more, they were in the same airspace than would be predicted by random. So it seems like another mechanism by which they are, maintaining contact with their group is that they may actually be foraging together, right? So yeah, I love that video to see them kind of flying around and doing stuff. Okay, so just conclusions from this section. Is that contact calls are playing a role in communication and group cohesion in this species. But honestly, there's still a lot of really interesting open questions in this system, not only like focused on that inquiry and response, but on these other types of social calls. They do produce other social calls that we don't know the function of. Okay, last section. Okay. I promise I won't go too long. So another big part of my research has to do with studying conservation of bats in North Dakota. When I first started in North Dakota, we knew very little about bats. There had been sort of reports over the years, like in the 60s, somebody caught some bats in a barn and published a paper about it in Minot or something like that. So my students and I over the years have invested a lot of energy trying to understand the ecology of the bats in North Dakota. And I didn't talk about them here because a lot of it doesn't have to do with the bioacoustics, and that was the nerd path I went down. So, but I do just want to recognize that, we didn't know a lot in 2009 when I started, but all of the people here have in some way been involved in gathering data about the ecology and behavior of bats in North Dakota. And I could give a whole separate talk on a lot of their research, but I won't. So I did just want to mention like why we care about thinking about conservation of bats in North Dakota, other than just like, they're non-game and we should care. White nose syndrome is a fungal disease that particularly attacks bats during the hibernation period when they're kind of like everything's shut down, including their immune response. It is fungal, and so you can see you get fungal growth on the nose here, but it also destroys their wings, okay? And this has been devastating to bat populations in the United States and Canada, where we see more than 90% decline of known winter populations, particularly in 3 species that are really hard hit by this. This is just to give you, we'll see if the video plays here, an idea of the spread of this disease. It started in Howe's Cave in Albany, New York, where it's believed it was brought in by a tourist wearing the same shoes they potentially wore at a cave in Europe where it's native. In 2019, it arrives in North Dakota. And myself and colleagues from other labs have documented white nose syndrome in really in central and western North Dakota. So this disease is here, and it can, as I said, have massive implications for the health of bat populations and can lead to huge declines, especially in a couple of species. Now, I'll just say that like 2 of those species that are really vulnerable to this, we have in the state. One of them, the northern long-eared bat, is federally endangered because of the impacts of this disease. That species is pretty rare in the state, so I'm not going to talk about them. But the little brown bat is currently being petitioned for listing under the Endangered Species Act because of massive declines in around the US and Canada. So if we need to monitor for bat populations to figure out if they are declining, and we need to make conservation interventions if possible, how can we do that? Well, when we study bats, there's basically 2 ways that we can assess what bats are present in the landscape. We can use mist nets. This is where the mist net is a very thin net that maybe you kind of stretch across a trail and the bats don't detect it and they fly into it, right? But mist nets are not really great for trying to assess population level trends on bats, right? Because you put a net up like along a forest trail and like the bat can fly all over the forest, right? And so you're sampling a very, very small amount of the space that these bats use, right? So an alternative tool that's been really important for monitoring relative changes in bat population sizes is bat detectors. I mentioned these are devices that can record high frequency sounds. You can put these things out throughout like an area, and they are really like standalone automated devices. They will automatically turn on at night. They will automatically detect and record the echolocation calls of bats. And then we feed them through programs that are able to identify those calls to species or species groups with pretty high confidence. And so these end up being a much better tool for monitoring bats across the landscape because they're not that expensive, whereas like catching bats, like that requires people who are vaccinated and skilled, and like it's a lot more intensive in terms of money and people than these things, which you can like just put all over the place, right? They're devices, they do their job kind of on their own without much human involvement. And so in North Dakota, we started a passive acoustic monitoring. So it's called passive because like nobody's out there listening, right? It's recording on its own. A passive acoustic monitoring study in 2019 that was designed to assess relative trends in bat populations across the state of North Dakota. And this was done in collaboration with all of these groups who either provided like logistics or funding. And each of these purple squares is one of our sampling areas. And you can see that they are spread across the state. And within one of each of these areas, we have 2 sites at which we put a bat detector. And just to give you an idea, I'm sorry, this is what each of these looks like. This is the bat detector here. This is like someone's farmyard, right? This is like types of landscapes we also get, especially in Eastern North Dakota. And here's the microphone that's kind of extended into an open space where the bats might be flying. So we collected this data across the state at 60 sites across the state. At each site, we would collect for 4 to 7 nights each year, and we tried to make those the same dates across years, so the data was really comparable. And this project involved a ton of students, right? I had one graduate student who did a master's degree with this work, and I've employed 10 undergrads across this 4 years to be driving around North Dakota and putting out bat detectors and such like that around the state. Now, because I'm about out of time, and I just want to tell you the major take-home message here, okay? We have 11 bat species in North Dakota. I'm only going to talk about one of them, that little brown bat, right? The one that is particularly susceptible to white nose syndrome, partially because we either didn't have enough data for some of the rare species, or the other species just seemed like there was not much change across the 4-year period. So there's that little brown bat. And I don't want to get too much into the methods, but you'll see on the next slide that when these calls are like acoustically identified, the program is like really conservative. And so it's like, and there's several of these species in the genus Myotis, which the little brown bat is, that have really similar calls, right? And so it tends to be really conservative. And instead of like assigning a call to like the little brown bat species, it's like, oh, we're going to bin it into the high frequency species, bin into the group, basically. But based on what we know about the distribution of this bat across the state, most of those calls in that high frequency group should be little brown bats. I only mention that because the next thing I'm going to show you is data that pools this, like, these are call sequences that were identified as either a little brown bat or a high frequency bat, which again, based on my knowledge, is mostly going to be little brown bats. And what you're seeing here is year on the X and the average number of files identified to one of these species groups per site per night. Okay. This is not good, right? So this is showing that we are seeing major drop-offs. In fact, a 72.4% decline in the recordings we are getting of these species across the state. Now, this is just pooled across the whole state, right? But overall, this is upsetting, but it's also not unexpected. This is what has been seen in other places that white nose syndrome has come through. And so hopefully this data will be informative to any game and fish who got it recently. And this will be something that can, mean that they really have to be dedicating resources to continuing to track these species and figure out if there are strategies that can be done to help them recover. Bats are very slow reproducers. They generally have 1 offspring a year. So, it's estimated that the losses they've experienced so far will take like 500 years for them to recover, right? Because they're just such slow reproducers. So I am ending on like a sad note. I'm sorry, but why I like to do that is because bats need friends, right? A lot of people don't like bats or have incorrect information about bats. And so I hope that all of you will be friends to bats if you were not already. And can, if you hear people giving bad information about bats, you can tell them about how cool they are. And with that, I will take any questions.
So maybe we'll see if this microphone works too. But we do have an online audience. So if you're online, feel free to put your questions into the chat, but we also can open the floor to anybody who would like to ask a question. And if I can, I'll try to run a mic around.
I'm sorry, I went a little long too.
I had a question about that last bit about the decline that you'd seen with the sampling. That decrease is definitely very dramatic. Is there independent evidence that white nose is increasing, or it's the cause of the decline? Any other evidence for that?
I mean, just because we've seen across the range as the disease has come, these populations have just been decimated. So like, especially in the Eastern United States, there have been studies where they have shown there are studies where it's been shown that like you go from like recording huge numbers of these files to almost none, basically. So it does just like fit with that and with the fact that there's just this spreading like disease epidemic, basically. So and you find dead bats too, right? Like you find them dead if they roost. A lot of times, like in North Dakota, we don't have a lot of like overwinter hibernators here. They mostly go to other places. There's some in the Badlands in Western North Dakota, but we think most of them leave. So like they could be in like Minnesota or South Dakota. And then like, you know, if they get white nose syndrome, a lot of times they just never come back. So yeah.
What are the chances that there could be an evolutionary response and there are few surviving, built back?
That's a good question. Craig's question was, what's the chance that there would be an evolutionary response and that the survivors would basically have, you know, the genetics basically to the right genes to survive this? There are people who are looking into that in terms of like what's left in terms of the survivors, because those populations are starting to grow again. I'm not aware that there's really been any like specific like that they've really found strong evidence for that. I could be wrong, but I know that people are looking at it, but I don't think there's been a lot of like strong evidence that these survivors are really like different genetically from the individuals that died. So. Roger?
I'm a fan of bats. Going back to echolocation, most of your plots don't do anything about directionality of ears or the vocalization, or perhaps these different ears.
Yeah, so Roger's question was about whether we consider directionality of the signal and that the ears themselves are directional in terms of the sound coming in. There are people that consider that for sure and think about that, usually more in a lab context. But there's people, you know, there's been a lot of work done both in the field and lab, like modeling the beam that's coming out of the bat and like how that beam changes. For example, they can change the width of the beam based on like how much they open their mouth, essentially, and also kind of those patterns as the sounds go into the ears. I will say that like those things get considered, but they tend to get kind of considered separately in separate studies. And we have like in that study I had done during my PhD, like we kind of looked into like Doppler effect and if that could potentially be driving the differences we saw. And we didn't find evidence that that was the case, basically. But it's a good question that these are the animal's signal is directional, their receivers are directional. But unfortunately, the field and the lab tend to be kind of two groups that don't integrate very well traditionally. So I think there are some people that cross that border well, but it'd be great if there are more. So.
Given how many talks you've done to the public, what's your impression of how people think about bats and their usefulness? Because let's face it, if you don't think something's useful and it's ugly to us individuals, most people don't care if it ceases to exist as a species or not. So do people actually get it in the state or not?
First of all, Dennis, they're very cute. Yeah, the question was about how people respond to a bats and like their thoughts on them and like how that affects how they see their value, right? And I will say there's a huge difference between adults and children, and I mostly speak to children, actually. So adults, they just, it's not like they're not open to ideas, but adults tend to have more fixed ideas about things, and they're less likely to like just take in new information and change like their emotional reactions to things like bats, right? Whereas children are much more open to that information. But it is problematic, right? Because how people feel about bats impacts like what they do if they find a bat in their house or something like that. And like, you don't want a bat in your house, right? But like, you don't have to beat it to death with a tire iron either, like, so yeah. And this is all really, the landscape of this has really changed with COVID too. So my graduate student, Lydia, she actually has done a study on the perception of bats and how that's changed with COVID, because bats are thought to be where COVID originated from. So in Asia.
So there is a question on the chat kind of related to this from Laura Devick, and she asks, are there any good resources for high schoolers to learn more about bats?
Oh, that's a good question. Where I would probably direct for information on high schoolers to learn about bats is Bat Conservation International, which is an organization based out of Texas. And they do generally provide a lot of educational materials across ages. So that would probably be a good starting spot. And they could at least, they're usually very receptive to like questions about that stuff. And if they don't have stuff, then they could certainly guide you to who would.
All right. Well, as we conclude, I think we would like to invite Provost Bertolini to make a few comments as we close. With the walking mic or the walking mic?
Thank you so much. Thank you so much for that great lecture. First, I want to say President Cook sends his sincere apologies for not being here, but he had an urgent legislative call that he had to be on. So he's sorry he missed it. I think everybody has a bat story. I lived a whole summer with a bat in a carriage house outside of Philadelphia. It was up in the stair hall. I'd just see it every day, and then it disappeared. Anyway, the Phi Kappa Phi lecture is one of the highest honors that we can bestow on individuals here at NDSU. And we're grateful for Dr. Gillam's research. We're a great university because of this kind of research, and we're grateful for everyone who participates. The other thing I would like to say about the lectureship that is so great is that, each of us know that, your particular research is kind of an area of convergence, right? That you keep focusing on a very small detail of life and become an expert on it. And that's what we find that we appreciate. But the lectureship actually opens it up to all of us novices, that we can actually listen to you and greatly appreciate why you're so great at it. So on behalf of President Cook, who can't be here, he sent the other David, I would like to give you this plaque to recognize tonight. And thank you so much for your greatness. Yep. Yeah, very good to see you. Anyway, I think we have a reception. Is it over outside here? So please join us for the reception out here. And thank you, Dr. Gillam, for your work so much. Oh, yeah, absolutely.
Okay.
I'll just drop the mic.
I do have some bats that I got last night, and I'm not supposed to bring them into the union, but I didn't ask permission. So, so if you want to see them, they're in here, and I could go outside and show you the one out there. See, there's one in there, at the top here. Yeah. So if anybody would like to see the bat, you can come down and take a look. So, all right, thanks.
Cute little bat in there. That's awesome.
Yeah.
Oh, let me go get.
There's one.
Somehow get a daredevil, superpower.
That's awesome.
Yeah, I got two last night, so there's one.